In the previous video, we talked about oogenesis, the female version of gametogenesis. In this video, we're going to talk about spermatogenesis and the primary sex organs, the testes. So this is a familiar diagram that I had displaying gametogenesis, specifically oogenesis, where we had oogonia turning into oocytes and then turning into ovum or eggs. In males, this represents spermatogonia, spermatocytes, and spermatozoa. Spermatozoa are synonymous with sperm cells. The big difference here is that this doesn't begin until puberty, whereas in females, it begins in fetal development. Here's a picture of a testis, and there's a number of different parts of this. In yellow here are the seminiferous tubules, where the production of sperm cells begins from spermatogonia to spermatocytes to spermatozoa. It is supported by cells within the seminiferous tubules known as Sertoli cells, which facilitate the growth and nourishment of the developing sperm cells. And between these in this white area right here are going to be another group of cells known as Leydig cells. And those are going to release the hormone testosterone. So those Leydig cells are endocrine cells. And we'll talk about the endocrine system at a later point in the semester. So let's just take a look at all the structures here. Once again, this testis is located within the scrotum, which is external to the body. So that's a big difference between females, where the primary sex organ, the ovaries, are within the peritoneal cavity in females inside the body. In males, the primary sex organ is external to the body, and it's composed of a number of structures, one of which are some protective layers. The tunica vaginalis is one of them that actually has two layers to it. An outermost layer, which is a parietal layer, and then it folds upon itself, and the innermost layer is known as the visceral layer. So the tunica vaginalis is composed of two layers. The outermost layer is the parietal layer. The innermost layer is the visceral layer. In between those layers, all the way around the testis, is fluid that helps protect and lubricate the testes. Once again, we talked about this seminiferous tubules, where the process of spermatogenesis is going to occur. Within dark orange here are the lobules. There's about 250 lobules per testis. And once again, the seminiferous tubules are in the testis. Between the visceral layer, which is this purple layer right here, let's look at it right here, purple layer right here, and the lobules is this white area, and that is the tunica albuginea. Tunica albuginea. Once the sperm cells have reached a significant stage of development, they are moved into an area known as the rete testes, where some maturation continues, and then via the efferent ductules into a region known as the epididymis, where the main time span of sperm maturation occurs. Sperm cells that leave the seminiferous tubules are not fully developed. They could not be fertilized if they were actually ejaculated directly from the seminiferous tubules. But those spermatozoa, otherwise known as sperm cells, complete development in this region right here known as the epididymis, and then during ejaculation, their spermatozoa, aka sperm cells, will leave this structure known as the ductus deferens and enter the pelvic cavity, where it will have fluid added to it and then become ejaculated out of the body. The ductus deferens is also known as the vas deferens. Okay, we'll continue on with male reproductive anatomy in the next video.